The title of these notes is Mutation and Genetic Variation. The connection between what we learned on mutations and what we're about to learn is that mutations actually create genetic variation within a population. A population is a group of individuals of the same species that mate and produce offspring. Genetic variation in a population increases the chance some individuals will survive and produce offspring that will also survive. Genetic variation leads to differences in phenotypes. Here we have two different phenotypes, a dark phenotype mouse and a light phenotype mouse. Natural selection acts on phenotype. Remember, phenotype depends on genotype and environmental factors. So here we see two different phenotypes of beetles. We see a green beetle and what they're calling a brown beetle. I think it looks orange. So let's say these birds really like to eat the green beetles. That means the probability that a green beetle is going to survive to produce offspring is lower than the so-called brown beetle. So what we see over time is an increase in so-called brown beetles compared to green beetles. Now, does that mean every single green beetle would get wiped out? Not necessarily. This is kind of an over-exaggerated example of natural selection. Now, the more different phenotypes there are, so remember phenotype is like the way you look, the more different phenotypes there are, the more chance for individuals to survive to produce offspring which will survive. And that's what evolution is. Two important definitions here. One, gene pool. A gene pool is all the alleles present in a population. Remember, an allele is a version of a gene. And remember, a gene is made of DNA. Allele frequency is the number of times an allele occurs in a gene pool. And we're actually going to be practicing calculating allele frequencies for populations today. Evolution, in genetic terms, involves a change in the frequency of alleles in a population over time. So here, this population of frogs has two different alleles, and they are represented by G's. So it has a capital G allele and a lowercase g allele. So the capital G codes for green frog, and the lowercase g codes for brown frog. So G and G, those are alleles. Now, what we're seeing here is that the capital G allele is dominant over the lowercase g allele, making the lowercase g allele recessive. Because you can see in a frog that has one of each of those kinds of alleles, it has a capital G and a lowercase g, even though it has an allele for green and an allele for brown, only the allele for green is showing up to make a green frog. So that is what we mean by dominant and recessive. A dominant allele basically overpowers a recessive allele so that the recessive allele is not expressed in the phenotype. Remember, the phenotype is what it looks like. But when you have two recessive alleles together, you get what we call the recessive phenotype. And in this case, for these particular frogs, the recessive phenotype is little g. To figure out allele frequencies, you just need to compare how many of the allele you're looking at there are compared to the total alleles. So there are 7 big G's in the gene pool, and we can count them. 1, 2, 3, 4, 5, 6, 7. So the frequency of the allele capital G is 7 divided by the total, which is all of the alleles added together. So there are 12 alleles, and we can actually shortcut that and not have to count every single allele by saying how many individuals there are and doubling that because each individual has 2 alleles. So how many individual frogs are there? 1, 2, 3, 4, 5, 6. So if there are 6 individuals in this population, there are 12 alleles. So we compare 7 divided by 12 for 0.583, and you can stop there, or you can multiply that by 100 to get 58%. So 58% of the alleles in the population are capital G. For lowercase g, we have 5. So we have 1, 2, 3, 4, 5. So 5 divided by 12 is 0.417, or if we multiply that by 100, we get 41.7 of those alleles are little g. Remember, alleles are a form of a gene, and we represent them with letters. And a capital allele means it's dominant, and a lowercase allele means it's recessive. And remember, dominant means whenever you have the two of them together, so like in this picture right here, only the dominant phenotype is going to show up, even though they have an allele for each type of phenotype, just the dominant one shows up. It kind of like masks the recessive one, is how you can think of it. We are going to do some practice, so the assignment you're looking for is gene pools and allele frequency. And we're going to start off talking about pea plants. So in pea plants, the allele for green seeds, green is dominant, so we're going to represent that with a capital G. The allele for green seeds, capital G, is dominant to the allele for yellow seeds, lowercase g. What we need to figure out now is the frequency of capital G and the frequency of lowercase g. So to figure out the allele frequency, you just take the number of the allele you're looking at and you divide it by the total of all the alleles. So maybe pause and write down this equation. Let's say in this population there's 50 seeds with 2 alleles per seed. That would be 100 alleles total. And the reason I chose that number is because your data here is given in percents. So if you assume there's 50 individuals, you know each individual has 2 alleles. So there would be 100 alleles in the population, and that just makes the math easier because each of these percents is saying how many out of 100 there would be. So let's talk through the data. We have homozygous yellow, so we know homozygous yellow is going to be little g, little g. So for homozygous yellow, it's 44%, so that means there's 44 little g alleles. For capital G, we're going to also look at the homozygous, because homozygous means that there's just one type of allele. So if it's green, we know it's capital G, and then because this is saying homozygous green, that means capital G, capital G. So we have 24 capital G alleles. Now, that is not all of the alleles because we have this whole section here of heterozygous pea seeds. So what you can do is just divide the heterozygous in half because that means within that heterozygous set of individuals, half of their alleles are going to be big G and half of their alleles are going to be little g. So if you divide this percent in half, then you get how many big G and how many little g. So 32 divided by 2 is 16. So that means there are 16 little g's and 16 big G's in this 32% here. So we need to add those to the other that we have from the homozygous. That's what we're going to do here. So remember, we're going to first figure out the frequency of big G. So that's the number of big G alleles, and we got to do some adding because we need to take the homozygous big G's and add the big G's from the heterozygous group to get our total of big G. So 24 plus 16 is 40. So our number on the top of our numerator here in our fraction is going to be 40. Then we need to also figure out the total of big G and little g. That was 100, and that's because we're assuming there's 50 individuals, 50 seeds, with 2 alleles each, so there's 100 total alleles. Now we're ready to do the math. 40 divided by 100 is 0.4, so your frequency of big G would be 0.4. Or if you multiply that by 100, 40%. Now, let's figure out the frequency of little g. So the frequency of little g is going to be equal to the number of little g alleles divided by the number of big G and little g alleles together. So we have 44 little g's, and we got that from our homozygous yellow because if it's yellow, we know it has to be little g, because that's the only phenotype that can come from little g. So homozygous just means it has the same alleles. So like little g is homozygous. So there's 44 of those. Then we also have to add the half of our heterozygous. So remember that's 16. So we have 44 plus 16 is 60. So there's 60 little g's. So that goes in our numerator, the top of our fraction. And we're going to divide that by the total big G and little g, which was 100, because remember, we said there was 50 individuals in this population. Each individual has 2 alleles, so 50 times 2 alleles would mean 100 total alleles in the population. Now you're going to do the same kind of idea with populations 2 and 3. For 2 and 3, to figure out how to set up the alleles, read that first paragraph at the top of your paper because it's going to tell you what's dominant and what's recessive so you know how to order the alleles.